Hi everyone and welcome to my talk, Detecting Genomic Regions that Determine Seed Quality of Brassica napus through Mapping by Sequencing. Brassica napus, also known as oilseed rapeseed or canola, is an important crop. This is mostly due to the high oil content. And therefore, rapeseed is one of the main oil crops in the world, only second to soybean. Germany is among the main oil producers by growing substantial amounts of rapeseed. You can see a flower down here on the left-hand side, and you can see the composition of rapeseed on the right-hand side. Of course, oil accounts for about 50%, and there's also substantial protein fractions. Then there are some other components, and one of them will become very important during this talk, the glucosinolates, which are among the off-taste components in rapeseeds. So today will be about mainly two different seed quality traits. On the one hand, we have a trade-off between the seed oil content and the protein content that will be investigated. So when extracting oil from rapeseeds, there is a protein-rich meal cake that is a byproduct. And this byproduct could be used, for example, to feed animals, but also for human nutrition. And it is important to know that the amino acid composition of the rapeseed protein is actually almost ideal for human nutrition because it's very close to the FAO recommendations. But there is, of course, a problem. There are many off-taste components in rapeseed that prevent the use for human nutrition. And among them are glucosinolates, the second focus of this talk. Glucosinolates are important off-taste components and are produced by the plants to protect them against herbivores and other pathogens. And of course, the GSL content was already reduced through breeding, but understanding the molecular basis of these biosynthesis pathways will help to improve the crops even further. So the objective of this project is to identify the regions that underlie the seed oil content, the seed protein content, and the glucosinolate content. And to do this, we use an approach called mapping by sequencing. The concept is illustrated here. We can separate plants into different pools based on their phenotype. So it's indicated here by showing orange and blue plants. And in a segregating F2 population, we can assign all the plants that show the phenotype indicated here as blue into one pool. And sequencing this pool, we can see that different genomic regions can come from the one or the other parent. But we can identify one region where they have almost 100% allele frequency coming from one of the parents. So then we can assume that this is a good candidate region that could underlie the phenotype that we are interested in. So we are interested in identifying such regions, and therefore we look for patterns in our rapeseed population. The workflow is indicated here. We have one parent that has a high, for example, oil content, and we have another parent with a low oil content. Both of them are crossed, and then the segregating F2 is analyzed. It's possible to assign plants in the F2 to the low pool or to the high pool based on the amount of oil that is detectable. Then DNA is extracted and subjected to sequencing. By analyzing the sequencing data, it's possible to analyze the allele frequencies, and then we can compare both pools because they should have similar allele frequencies at most genomic positions, but we are looking for the few positions where they have substantial differences. And you can see it here in the example. We are trying to find a region where high pool and low pool differ substantially. And these regions are candidates. And by using this approach, we identified genomic regions that are likely to determine the seed oil content and the seed protein content. For example, you can see here all pseudochromosomes of rapeseed, and we see a strong signal on chromosome C8 down here. And this region contains the gene phospholipase that was described previously and might improve the oil stability. So it is a potential explanation for the region that we see here coming up in our analysis. Some of the other regions also align with the literature, and we are currently looking into these regions to identify individual candidate genes that might explain the trait. Here you can see the results for the glucosinolate analysis, where we try to identify genomic regions that are responsible for high or low content. And we see a strong signal on chromosome A9, on chromosome C2, and on chromosome C9. And these are regions where key regulators of the glucosinolate biosynthesis are located. And again, we are now looking into these regions in more detail to find the individual genes that are responsible for high or for low GSL content. It is interesting to know that this region on chromosome A9 is rich in large structural variants, so this might be associated with the phenomenon that we see on the phenotypic level. And this brings me to the end of my talk, and I would like to acknowledge the great support from a team that made this project possible. And of course, I also would like to thank the BMBF and PTJ for funding the project. Thank you very much for your attention.